Now, from the law of mass action, we learned that the energy that we can derive from a chemical reaction, for example, from ATP to ADP, depends on the concentrations of both the reactants and also on the educts. Now, there is another concept that is also coming from the law of mass action and reversal reactions, and that is important, equally important in biology, and relates to the charge of proteins. Now, what you see here is a protein where the charge on the surface of the protein is marked in colors. So in blue you see the negative charged protein patches, and in red you see positive charged protein patches. Now, the interesting part is that if you change the pH of the environment, the charge on the protein will change. And you can imagine this has a tremendous effect on workings of proteins. If, for example, the interaction between two proteins is dependent on the charge, that means depending on the local pH of this protein, which might change also in the due time course of the cell, the reaction will take place or not, or interaction will take place or not. Okay, so the summary here is of that graph is proteins can be charged, and the charge depends on pH. Well, the chemical reactions that play a role in protein charge are actually two chemical reactions. The first is called the protonation, where you put a proton onto a protein and that by that change the charge to the positive side. That typically acts on amine groups. So amine groups are groups which a lot of proteins end, for example, with an NH2 group. And you can add to that NH2 group one additional hydrogen ion, leading to an NH3+. Likewise, there's also deprotonation happening. That means you split off a proton from a protein or from an amino acid, and that happens typically at carboxyl groups. So carboxyl groups is actually on the other side of the protein, which is ending with a COOH, and this hydrogen you can split off, leading to a negatively charged COOH and a hydrogen which is then dissolved in the environment. Let's just, for an example, just consider the deprotonation of carboxyl groups. Well, for this chemical reaction, we use the law of mass action, and we can derive the pKa, so the log of the equilibrium constant, is equal to the log of the concentration of the COO minus groups times the concentration of the hydrogen atoms divided by the COOH groups. Now it's interesting to calculate, so what is, after the reaction has taken place, what is the probability to find a deprotonated protein? Let's call this probability P, capital P. So we want to know what is the concentration of COO minus groups as compared to the total of COO and COOH groups. So we divide by the sum of the concentrations of the two letters. Now, that we can calculate just by the pKa. So we divide both sides by the concentration of CO minus in order to obtain that this probability is given by 1 divided by 1 plus 10 to the power pKa times 1 divided by the concentration of the hydrogen atoms. Now we have to realize that the concentration of the hydrogen atoms is given by the pH of the environment. Hence, the probability to find a negatively charged protein is then given by 1 divided by 1 plus 10 to the power pKa minus the pH of the solution. Hence, this is exactly what we wanted to show. We understand the net charge of the protein depends on the pH of the environment. Now, what people typically define is the so-called isoelectric point. This is the point where the pKa is equal to the pH, and that means that in this situation, half of the carboxyl groups are deprotonated and half of them is not. Here I show a list of all pKa values of all amino acids. And you see that the pKa value for the carboxyl group is in the order of between 2.3 and 1.8. Likewise, you also will find the pKa values for the amine groups and also for some other residues of amino acids. Further, I copied here an experimental result where the number or the concentration of the hydrogen radicals is displayed as a pH of the environment. Now, taking all the pKa values and all the residues into account, we derive at a kink in this curve, which displays the so-called isoelectric point. This is the point where a protein is uncharged. Now, we argued that the charge of a protein might be of tremendous importance for its function. Maybe it cannot even recognize its partner and can't bind. But it also has an important technical implication, because it gives a handle in order to separate different proteins. And that is called, the method is called protein separation by electrophoresis. So what you do in electrophoresis, you have a mixture of proteins and you load them into small buckets, here 1 and 2, which are located on one side of a gel. Subsequently, you add a voltage towards the other side of the cell. And what you will find is, because proteins are charged, they will move through the gel. Now, what is important is that on the gel, the speed by which each protein will move depends on the mass, but also on the charge of the cell, and is characterized by the so-called electrophoretic mobility, or with other words, the speed by which proteins are moving. Now, the electromagnetic phoretic mobility we can understand from the Einstein relation. The Einstein relation makes a relation between the speed of a particle, a force that is applied to the particle, which is in our case of electrophoresis is Q times V, so the charge of the particles times the voltage, divided by the friction coefficient. Now, if we assume, and that we learned in physics of life 1, that proteins are basically spherical particles, we can assume Stokes' law, and Stokes' law makes a relation between the shape of the particles and the friction coefficient, and we learned that the friction coefficient is proportional to the radius of the particle. And knowing that particles are spherical, the friction coefficient scales like the third square root of the mass. Hence we see the speed by which proteins move on a gel is given by the ratio between the charge and the third square root of the mass. In this way we can separate different kinds of proteins. And the method is called protein separation by electrophoresis. So today's take-home messages. For the description of biological systems, we should use the canonical ensemble. Equilibrium in the canonical ensemble is described by a temperature and the chemical potential. We derived an equation for the chemical potential of an ideal gas and found that it's a sum of two parts. The first describing the concentration of a substance, and the second the standard chemical potential, which depends on internal energies in the system of the particle. From there we were able to describe one of the main laws in chemistry, which is the law of mass action, and defined the chemical equilibrium constant that is important in order to understand, for example, charges of proteins, but also the energy that is available to a chemical reaction.